Today we have with us Professor Nagasuma Chandra from the Department of Biochemistry, and she is here to talk on the topic, Can Biology Be a Predictive Science? So, Professor Nagasuma Chandra, she was one of our instructors for the UB 101 module on metabolism, which I'm sure all of us liked and we thoroughly enjoyed her course. And she's a distinguished professor and a Purna Pragna research scientist at MBU. She is all, but still, which is a yes. So she's also an associated faculty member at the DST Center for Mathematical Biology at ISC. Her work involves various aspects of computational biology. So she works on systems biology, drug discovery, structural bioinformatics, and computational immunology. All of them are on the frontiers of biology research today. Being an eminent researcher, she has taken out time from her tight schedule and she's here to talk to us today, and which we really appreciate, ma'am. And so she's here to present a computational perspective of biology, and seeing the interdisciplinary audience we have here, I'm sure we all are going to enjoy it. So please welcome Professor Chandra onto the stage.
Thank you. Okay, it's my pleasure to be here, and I hope I won't disappoint you in the next hour. So I want to share my perspective on what biology can be. Okay, most of you are familiar with the common practice of biology, right? Prediction and biology don't go well together at all. Most biologists, in fact, are still skeptical about prediction. Okay, but I hope in the next hour I can convince you that need not be the case, and then why and how we can do prediction and how we can benefit from that, especially for understanding diseases and diagnosing, curing perhaps, and also preventing to some extent. So that will be the focus. So why is biology so different in the first place? Okay, so biology is different because it's an outcome of a random tinkering process, which is nothing but evolution basically. It's not something that has been built with a blueprint, right? Unlike our engineered objects, an aircraft, a car, a computer, or your radio, whatever that may be, right? That has been designed with a blueprint. You get exactly the same answer. In fact, it's all based on predictions. People don't design a car, rip it apart, and then see whether it's working or not, and then go back and do that. That's all done. The science is well understood. It's become technology now. So it's a very predictable process. So you don't get completely different answers. Of course, there are challenges in every area. That's not what I'm referring to, but by and large people understand what they're doing. In biology we can't really do that. We have some success with synthetic organisms, but do we have a way by which we can design a super bullet, a magic drug for example, to cure a particular disease? Well, the answer definitely is no. Okay, so let's see why it's so complex. Again, it may not be so very difficult to understand that. Worldviews of three different perspectives of science, right? Physics, chemistry, biology, of course maths is part of everything. Now let's look at building blocks in physics, right? You can explain many things perhaps with the elementary particles, the 28 elementary particles, and everything is built over that and well understood and so on. Okay, in chemistry, of course, again, what is relevant to biology is what I put here. There's a whole lot of other things. Again, maybe thousands of building blocks of some kind, hundreds to thousands. The numbers are still somewhere around there. Whereas building blocks in biology, at what level should you consider as one thing? First of all, if you look at the species, they look very different from each other, as you will appreciate, right? At least 10 to the power 7 species at the level at which they're classified. It could easily go an order or two of magnitude higher, depending on how you characterize whether two different strains or two different species, or where the definition lies is a question. In any case, it's a very large number. Okay, now what about the different cells, right? In humans, there are trillions of cells, 10 to the power 12 cells. There are many cell types, hundreds of cell types. The numbers are very high and their location is different. So they're different from each other. There's some common threads, but they're still different. Now within each cell, what's the level of complexity we have, right? We know each cell has a genome and it codes for many proteins, and different types of proteins themselves can run into 10 to the power 5 species. 10 to the power 3 to 10 to the power 5, depending on what organism you're looking at. As if that was not enough, you have different forms of each one of these. So what I have here is this the pointer? Okay. Stick, okay. I remember my adventure with this. I don't know if some of you were there in the first class. Anyway, so we have different protein structures, what I represent here. There are at least 10 to the power 5 protein species in a human cell. And as I said, as if it's not enough, each one can exist in multiple forms, an R form, a T form, a phosphorylated, a dephosphorylated, conjugated with a sugar, a lipid, or a protein-protein complex. So it goes up higher and higher. I don't think we have a count of how many different types of protein species exist at a given point in time in a cell, but it's much, much larger than 10 to the power 5. Another layer of complexity, the counts for each one of them can be very different. Hemoglobin can exist in millions, whereas some other protein may be in hundreds or thousands. The number of copies of each one of these in a given cell can be very different. And now you put them together, so many different cells, different locations, gene expression level varies. So it's a very, very complex thing. The complexity is pretty large here. So it's not surprising, therefore, that the language of physics, which is reasonably well understood, right, is boiled down into perhaps a complex equation, but then can be determined, right, can be solved. So you can get an answer from a given initial conditions. Ditto with chemistry, okay. But in biology, the language of biology is more long text like this. I'm sure most of you would like a true or false or an objective type question which you can't have a second answer for, but unfortunately that's not the case. There's always some justification for something else. So it's quite complex. Why? Because it's context dependent. You can't have a single answer for a question such as what happens if a particular receptor is phosphorylated, right? At best, you can make mental maps or maps that have been derived from laborious work or for decades perhaps. You can make pathways which are complex. You'll get some structure. It's still very, very complex. Or another way of looking at it is like this, a jungle of things interconnected with each other, which we call as a network, right? And each dot there can refer to a protein and how proteins talk to each other, whether they're all connected. It's an orchestrated whole that is life is all about, okay? So biology versus other disciplines, the main difference will be scale and complexity, diversity, okay? The diversity in terms of number of species, and the variation, the diversity is also intertwined quite heavily because of the ability of living species to adapt to their particular niches, that ecological niches that they're looking at. And therefore, it has to be high. By definition, the diversity incorporates this kind of changes that are there. So as a result, you can ask, in physics, what happens if I drop this ball and get an absolutely accurate answer, right? You know exactly at what speed it will hit the ground and what's the impact, and you can calculate the impact on the ball, on the ground, all sorts of things you can do. You can do perhaps more or less similar things with chemistry. If you have an initial conditions, you mix A with B, can you say how much of C will come? What is the, what is the rate at which it happens, whether it is driven in the forward direction or the backward direction, if at all it's reversible. It's pretty predictive. And in biology, it is not so, because if you ask a question, what happens if TGF receptor is phosphorylated, you get answers such as this, which is long, and if this is there, and if that is not there, and if something else is there, then maybe this happens, that kind of an answer. So it's context dependent, because a given protein or given gene can perform different functions depending on other things around it, okay? So as a result, traditionally biology has been an observational science. The observations have got more and more and more sophisticated. We are able to manipulate cells, we are able to manipulate genes, we are able to manipulate so many things. You have the latest CRISPR-Cas technology, you can do a variety of things. You can insert a gene, you can knock out a gene and see the effect and so on. But it's still stamp collecting at the end of it. Okay? And they're extremely useful. By no means am I trying to be derogatory here. Without these stamp collections, we cannot even think of proceeding further. The stamp collections have changed their landscape in that sense. They've gone on from the traditional thing that you've seen from Darwin's days on to now genome sequences, a whole lot of sequences, now cellular images, protein structures, various types of images at various levels. Okay. So is this really sufficient? If you look at a disease like tuberculosis, we have a whole lot of, again, stamps, data basically, data coming from clinical data, the microbiology data out there, and both for the host and for the pathogen, we have a whole lot of layered data available here. So is this sufficient for this to become a predictive thing? What is the challenge basically? If we collect a whole lot of data, is it sufficient to say that you have understood the whole thing? The real challenge lies in interpretation of this data. What do we make? How can we make sense of all of this? How many of these, for example, the Mycobacterium tuberculosis genome codes for about 4,000 proteins. How many of these 4,000 are absolutely essential for this bacteria to cause infection? Essential for it to survive and for it to cause infection? And is that the same in two different individuals? These are the questions one can ask. And the other side, you can think about the host. What in the host makes somebody more susceptible to your disease as compared to their family members, blood relatives, or neighbors, or anything like that who are exposed to the same atmosphere? Okay. In fact, you have detailed repositories. Now, this is a biochemical pathway chart, which most of you would have disliked studying, right? Okay. Anyway, so each one, can you recognize this? Each one here is a metabolite, and each dot, if you see here, is a reaction, a biochemical reaction that's going on. And at any given time, every second of our life, in fact, 400 billion or so reactions go on. Okay, every second. This happens in a very precise manner, repeatedly, all the time. A whole lot of these reactions are going on. And I should emphasize that it is a highly oversimplified image. Okay, this is an old textbook image. We have much, much more data now for all of these things. At least this should tell you why we need to handle, why we need to do, understand this, why we need a computer to do that. But that's not all. It's not just the scale that is, asking us to go into computational methods. It's actually the need to interpret this, as I'm saying. So the grand challenge of today actually is to make biology a predictive science. Prediction not because we like astrology or something. Prediction is very important because if we understand a system, we should be able to predict what it does, its behavior, right? In fact, it's often said that you don't understand an object fully until you take it apart fully and reassemble it in exactly the same way. Both in form and function, you should be able to understand what's happening. Therefore, prediction becomes very important, right? And this is a huge paradigm shift that's out there. From data, of course, data has been coming in unprecedented rates, basically. The data to models, now what sort of models we need, and it generates hypotheses, and finally it should give us theories. Much of biology research has been hypothesis driven. Hypothesis, again, needs to be understood in context because it can have different meanings in different contexts. Hypothesis is you have some sort of an idea to start with, and then you test that idea. That idea is preconceived. It can come from somebody else's experiments, it can come from some other prior, educated guess of some kind. It can, variety of reasons how hypothesis can come. But does it mean that this is the best hypothesis for that particular question? Okay, so the answer is really no in many cases. It may be, it may not be, but it's still unexplored. Much of it is unexplored. To give you an example, you pick a particular gene and say this is the most essential gene for this bacteria, and I want to inhibit it, and I want to make a drug out of that. Okay, so that's a hypothesis. That particular gene, it can be a very important enzyme, that makes, say, the cell wall proteins or something like that. But is it enough as a hypothesis? What else happens? Can the bacteria now adapt when this particular gene or its product that is a protein is inhibited? What can it do? Can it overcome that in some form which you have not really understood? Drug resistance is a major problem. Drug resistance comes because the bacteria is not a dumb thing. It is adapting. It has the ability to adapt by changing itself in many ways. Okay, so that's what evolution is all about anyway. So we want to actually generate hypotheses that may be most suitable for a particular thing. Okay, so there are two ways people talk about this. One is a hypothesis free, in the sense you don't really start with anything, still analyze the data. This is possible only through computation, and today it's the word big data is used. We'll not get into the debate of what's called big data. In any case, very large data is available and how best you can interpret this. No? Again, there are two ways. One is a machine learning approach, as I'll tell you. Another one is a model-based approach, which you understand something about. So you get models and hypotheses out of this. You test the model, and finally it becomes a theory when you have verified it. So can you think of any theory that is fairly well, a standard accepted theory in biology?
At what level we can
any level.
Evolution, cell theory, we can go.
There are many, right?
Genomes, and then what you call it.
The genetic code is a good example, actually. The standard genetic code, you don't question. You don't try and predict. You don't try to see if there's something else other than a nucleotide that is going to code for your amino acid, right? So it's become so standard. The DNA double helix is another such kind of a theory now we can call it. You understand replication. There's no other molecule or some other machinery that is the basic unit of this particular process, right? So there are generalizations of that kind. Although in a discipline like physics, if you look at it, theory is much more respected and believed than the experiment. If there is a discrepancy between theory and experiment, it's quite often the fault of the experiment, and they're asked to go back and verify it. But in biology, it's the other way around. It's always, it's most often it has been like that, although it's changing quite extensively. Why do you, first of all, need a model in biology, right? It's so diverse, it's so complex. Do you really need to do that? Is stamp collection not sufficient as long as you address the problem? Is a question one can ask. Because it's quite obvious if I tell you these things, why modeling, simulation, and theories that emerge from that become very important, right, in biology, as it is in any other science. Okay? So, in fact, I personally believe that it ought to become, and it will become, an integral part of all biology, right? Whether it's evolution or microbiology, or whether it is disease biology at any level, or any biotechnology process that you want to look at, it should be an integral part. What are the advantages now? Okay, just to kind of put them into a formal context. It gives you a precise definition of the system components. You all understand the terms genotype and phenotype, sure, right? So what is the input, basically? The genotype, A, B, C, D, whatever that is, a precise definition of the system components. And it enables simulation in the sense you can predict a phenotype. If you have A, B, C, D ingredients, what sort of a phenotype you will be getting? That kind of a question you can ask. You can analyze perturbations of various kinds, in the sense you can do a knockout. You can change parameters systematically, one by one at a time, or in pairs, or in triplets, or whatever power your model has and your computational method has. Based on that, you can do a complete scan of the parameter space and then arrive at a particular parameter that is a controlling parameter for that particular process. Okay? So you're familiar with rate controlling steps in biochemical pathways. So if you model a biochemical pathway with their rates, now it's like various cars that are there going at different speeds, and then of course here the analogy is different because one car becomes another car, another car, another car, and finally you get a product, correct? So at the end of it, you want to know how much of which car has accumulated at a given point in time. And if you were to use it in, let us say, a biotechnology process where you're manufacturing something, you really want to understand which is the optimal set of inputs that are required, and what is the optimal order, and so on and so forth. So you want to make perturbations. You want to knock off something or knock down something and see whether the final product is still coming. And by doing that, you actually understand the controlling points of this pathway. Just like, painstakingly people have determined which is the rate limiting step or the controlling step of a biochemical pathway. It's just one example. You can do this for a variety of things. So you analyze perturbations. It allows us to predict, model, and conduct experiments that are beyond current capabilities in a computer. Okay? Now, I'll give you another example of tossing a coin. Right? Is the outcome of a coin toss predictive? Most of you will say, no, if it's a fair coin, let's assume we are good people. Okay. Well, it can be if it is in a controlled atmosphere. If you ask a robot to do it in a very precisely controlled manner. So the genotype for tossing a coin, if you look at it, can be a variety of things. The pressure at which you flip it, the weight of the coin, the air pressure, various things that you can think of, you can put it in place. Then you should get an exactly same outcome all the time, right? So that's the genotype, and now that's the phenotype for you here. It also gives us temporal and spatial meaning. Temporal is over a period of time, and spatial in space, in 3D space, of hard to understand processes, right? It can even be something like how a tumor spreads, for example, over a period of time. What is the, is it a fast growing tumor, is it a slow growing one, and why, what are the reasons for that, and so on. In principle, it should be possible to model all of that, right? We may not be there yet. I don't want to give you an impression that everything is understood and so on. We're just at the beginning, as I'll tell you as we go along. And it also gives us an opportunity to see what's too small to be seen. Now, what's too small here comes again in the context of our experimental capability. Too small is something beyond the resolution of the particular experiment that you're looking at, right? So you can actually think of doing this. Then you can, of course, these are more obvious advantages: designing minimal or efficient system, cost, and high throughput analysis, the speed at which you do things, and so on. But nevertheless, modeling becomes very useful. Now, how do you go about modeling? What do you model? Let's say I want to model an infection now. Where do I start? Okay. Ideally, you would like to understand everything at the most detailed level possible, right? You all understand the hierarchy of living systems. A whole cell is now made of, you can have an organism into tissues, or even from a cellular level, you can go down into, say, different assemblies and then proteins. And what is the highest level of anatomical description at which you can describe any living process? It's at the three-dimensional structure of the protein molecules, basically. Atomic structure, you would like to understand that. And you would actually like to understand something at the level of Schrodinger equation of where in a given atom the electrons lie, and can you calculate that, and so on, and then take it all the way upwards, right? In fact, this is called a bottom-up approach, but it's not possible. It's not beyond our capacity today to, even if you were to do it for one or two things, you can't solve beyond, say, hydrogen or helium. You can't even solve it for glucose. And if you were to do that, how do you make use of the data to the next level and the next level and next level? It's still a challenge, basically. So you need to be realistic, basically, although you would like to do it at that level. So it is not possible. So people try and do two types of things. One, you see, the resolution of information goes down this way. Now here you have a cell and you have a whole lot of data, genomics, proteomics, sequences, and so on. I will touch upon this as we go along. And you need to have a mathematical way of representing this, and a computational way of simulating all of these things, and that's the crux of bioinformatics of various forms that we come across. So having said that it is very difficult, very complex and all that, there are ways by which we can structure this complexity. In fact, we learn from engineering of how to do this. And structuring complexity, we can simply mimic the natural hierarchy that exists in biological cells. So you have a large-scale organization which is comprised of functional modules, regulatory motifs, metabolic pathways, and genes, whatever proteins, metabolites, all of these things at the fundamental level. And even this is across information storage, processing to execution, the same kind of structure, complexity can be structured. So we need to capture these various layers in various forms and then have something that flows through the various levels. So there are two approaches. One is the bottom-up approach, like I said. So you start from the basics, the building blocks, to the level that you can define. Right? You can also have a top-down model. In fact, most of the epidemiology models, right? People predict how fast a disease will spread, or whether it is spreading, or whether it is a high-risk zone or a low-risk zone, and so on. So those are called top-down models. Now, this is somewhat similar to the models that predict how the stock market will change, perhaps. It's a top-down thing. You're not basing it on some physics and starting from there. It's more data-driven. More from the trends, basically some sort of modeling will be done. Models are very, very sophisticated, but they're not detailed. So there's a big difference between the two. Okay? So you can have a bottom-up model. So at the lowest level, it depends, of course, on the data you have and the question you have in hand. Right? So you have different models. A model is what? It is something that will mimic a real process. Right? Just like a mouse is a model of a particular thing that you're studying, or a cell line is a model of a particular tumor that you are studying, a computational model is also a mimic of a biological process that you are trying to understand, basically. So it depends on the data that you have. Now today genome sequences, as I said, we have a large stamp collections, we have genome sequences available, it's filled up many computer disks, not books but computer disks. So we start with sequences, right? Again, I assume that all of you are familiar with what a sequence is. If anyone needs a clarification, please raise your hand. Okay. Now sequences, these are the molecular components. So what's the model we have here? We have a sequence to function model. In the sense, if a sequence is something like this, then this is the function that we can predict from that. And how does that work? It happens by comparison with known sequences. And comparison itself is a challenging thing, right? And we also believe that there's commonality through descent, that is evolution for you. Because two sequences are common, whether it's E. coli and something or whatever that is, we say that in function of one of them is known, we can translate the function, transfer the function from one to another quite easily. Okay? In fact, most of the databases that are available do it in that particular form. Now, whatever you can do with sequences, you can do it much better with three-dimensional structures because they're more telling. Sequence is now, think of a lock, right? Think of a thread that would have made a lock. Think of some chain, chain of beads or whatever that may be, that's made a lock. Now the chain of beads is a sequence. How exactly this chain of beads is folded or onto itself in a precise three-dimensional manner is the three-dimensional structure for you. So that is more revealing of the function. It's also more conserved in nature because at the end of the day, the evolutionary constraints are on function and not on what has given rise to that function. Correct? So 3D structures become absolutely important there. So like you can have sequence to function models, you can have structure to function models as well. And you can have models at different levels. Suppose you didn't have this kind of data, but you have some understanding about the pathways, or you, in addition to this, if you have some information about that, you can have biochemical pathway based models. If this pathway is present, then this bacteria or this particular cell can live with this particular carbon source as a nutrient, right? It can grow with that or it cannot grow with that. These kind of questions one can make. And it goes all the way up, so I will not take you to that. So somewhere here, cell is a fundamental unit of life, as you'll all agree. So many times we, because of the complexity and because of our inability to go further through a bottom-up approach, we may just aim for understanding at the cellular level. At the top-down approach, you can have, like I said, disease epidemiology and various other kinds of things. And today the boundaries are quite blurred. In fact, this used to be called theoretical biology, okay, and whereas this is genomics or downwards bioinformatics basically. Somewhere the combination of the two, the merger of the two, if you like, is systems biology, which spans across all levels. Again, it's practiced in a variety of ways, basically. Okay, now the genome sequence again is compared to your book of life. I won't go into detail about the analogy. It's not very difficult for you to think about it. It's except that the book doesn't have a particular way of writing. It is not necessarily from left to right. Some things are from left to right, some things are from right to left. Some of the letters in this book do not make any sense at all. Okay, so you do not know which makes sense, which do not make sense. I'm talking about coding regions, non-coding regions, and so on. Okay, and I'm also talking about whether it's 5' to 3' or 3' to 5'. Okay, so it's a very complex thing, and this book comes without any punctuation whatsoever, right? You're not even sure if everything is in English, so that's another trouble. Okay, but again, there are many ways by which you do that. Now, there are a whole lot of databases that are available, which stores these sequences, which we actually consult quite often. We do a similarity search across. If I have a new protein that I have discovered as important, or let us say it is upregulated in a particular cancer. Now, I want to understand what this does. It may be just named as a hypothetical protein. So to understand this further, we compare across whatever that is known in databases, which are fortunately accessible to everybody. I think it's been a big change or a big driving force for us to develop computational to do that, the easy and free access of data across. Okay. Again, this just tells you that are we justified in doing that? Is a question that I was going to ask. Yes, indeed, because common design principles underlie diverse expressions of life. This wants me to go faster than this. In any case, okay. So I'm sure you've got the gist of it. So how do we do this? So bioinformatics is a discipline which really helps you to do that. Again, it's defined in various ways, One of the best definitions of bioinformatics that I like is the goal of bioinformatics is to conceptualize biology in terms of life, conceptualize biology in terms of physicochemical principles using mathematics, using computational methods that are available. So in order to understand all life processes at the physicochemical level. So that's the main goal of that. And what helps us is all this omics data. I'm not a great fan of the jargon, but for lack of a better term, we do stick to omics word. It simply means that the coverage is as complete as possible. Genome is a set of all genes in a cell or an organism, whatever. Similarly, the proteome, the set of all proteins. Well, all would be a lie, honestly, because we'll never get all. So it's as much as possible, you know, that we're reasonably happy with that. So you have 3D structures, whatever people use, all these terms, simply to reflect that they are looking at a genome scale, you know, a genome-wide coverage of these various things. Okay. Now, the bioinformatics has changed. In fact, as I've seen in the last 15 years, tremendously, significant changes in our own goals, okay? Not just in one little corner of the world, but everywhere for that matter. Definitions of this science are being rewritten and boundaries expanded. What was narrow has expanded, and now it's encompassed quite a lot significantly. Generated a whole lot of hypotheses, new ways of research and application opportunities have come. Okay, and today the challenge and what is possible is scaling up from molecules to cells to whole organisms, yet retaining whole information. Since we want to reach that big thing, but we do not want to lose sight of the detail that we have, because the beauty of biology is in its richness, is in its complexity, and therefore it's in its detail. Okay. So in fact, in this prism, which is data to information to knowledge, see, data when it's processed becomes information, and then information is analyzed, validated, and interpreted, it becomes knowledge. You can take it further on and then call it wisdom when you're absolutely sure of what it's like. We are still at the lowest, this has moved for some reason, it should be somewhere there, we're at the lowest height of this knowledge peak, and we still have a lot of height to scale. Okay, what sort of algorithms are available now? So first and foremost, the human genome sequence, you know, the assembly happened because of really beautiful computational methods to put them together, right? I won't get into how the sequencing is really done, but without this very nice method that people came up with, and without the computers that were, you know, designed just for this purpose, we would not have finished the sequencing of the genome ahead of time, we in the sense, whoever did it, the community basically, ahead of time and with lower cost than planned. Okay, this is one of the landmarks of the century, in fact. So we have a whole lot of methods that are available for comparing now gene sequences and protein sequences. Proteins are better for many reasons. Okay. Now this sequences are like a sentence, an English sentence, only we don't have 26 letters in the alphabet, we have only 20 here, right? And this 20, of course, just 20 amino acids are enough to create the whole lot of the repertoire of proteins that we see in all life forms, right? So that means there's a lot of sensitivity that is necessary in comparing them and then analyzing them and so on. So a lot of text matching algorithms have come about, string matching they're called, and unlike comparing to English sentences, we want to also have the ability to say R equals K when we are comparing proteins, because R standing for arginine may do the same job as K, which is the lysine, right? Or S and T, for example, serine and threonine and so on. But that doesn't happen all the time. Not in all situations is an arginine capable of replacing a lysine, right? It's dependent on the position in a particular protein and so on. So there are a whole lot of considerations that one need to have. Okay, and a set of algorithms called dynamic programming methods. Again, I won't get into the detail in the interest. So which helps you, put them in a pairwise fashion first. If I have this as one sequence, what is the best way by which I can align? Can I align at all? Is there some similarity which tells me that they have some common ancestry? That is homology. The term homology means there's common ancestry which necessarily has to have some similarity, and therefore you believe that there is some commonality in function. Okay, so this is all. So what you actually measure is statistical significance, but what you want to infer is biological significance. So you need to be very careful about this particular thing. So from pairwise, now each one is a protein from a particular species. I show only a small portion. So one line here, the second line here. From that, you can go on to multiple sequence alignment. You can take this sequence, do a BLAST. BLAST has become a verb now, which is an acronym for Basic Local Alignment Search Tool, basically, which means you can just go to the databases, the NCBI database, and give your the protein of your interest, give the sequence, and see what else is similar to it. A whole lot of things. You can predict that they're similar. You can predict their function. This is a sequence to function model, basically. The function you can predict at different levels. You can definitely say whether it is now a protein kinase, because it is similar to so many other protein kinases. You can also say where is the site at which it can phosphorylate some other protein. Is there a motif that is conserved, and so on. You can tell all those kind of things. At the heart of this are substitution matrices, the way by which to compare, and so on. There are new methods that are coming about, but there are many methods that are already known. You can extend this to study domain architecture of individual proteins. Proteins are modular. They're multi-domain in nature. This may be a domain, another domain, another domain. Whenever I see a domain, that means it has a particular conserved function. Maybe an ATP binding function, right? Just by looking at this portion of it, which is much more sensitive than by looking at the whole thing, I'm able to tell what sort of function that is. So there are methods available. Some of them are so standard that you don't need to know any coding. You can just do a search. You can just trust the methods that are there and then do that. And at this level, it is an integral part of all biology. You go into any lab, either here or anywhere in the world, they will be using these kind of methods to understand the genes and proteins. Okay. Now people have looked at the whole organisms, right? You can compare now mice and men. Well, it's not gender biased by any chance. It's the title of a paper. Okay, here, right? Okay. So you can compare humans to rats and mice, whatever you have here, R and M compared to human, and all this 22 plus X or Y chromosomes. Wherever you see a similarity, the bar here. Okay, but the colors are based on what it is like in human. So what you see is the chromosomes are all filled up everywhere. That means there is some counterpart of this particular gene in the rats and the mice. Okay, but the colors are jumbled up, which means it may not be in the same chromosome. It's somewhere or the other. So as a pool, as a bag of genes, if you look at the similarity, it may be 98%, 95%, chimps and humans, 99, whatever, the percentage doesn't matter. But there's a lot of difference in the phenotype, as you will agree, right? And the differences may come from this jumbling up, their regulation and how that happens and so on. So synteny is a term that is used to look at the order of conservation. Okay, this has been taken further now as a predictive model to understand tree of life and the evolutionary clock, right? How do different organisms compare with each other? First of all, you have to rationalize existing data. Any predictive model has to rationalize, observe, you have to be happy and validate, and then it becomes a predictive model. And the rate at which mutations take place and so on, the evolutionary clock is a kind of a question that people ask. It's pretty important in now looking at viruses, how viruses evolve, what is the recombination rate, how fast do they change, or bacteria for that matter. Maybe very important in terms of what new strain may be coming out, what new influenza H1N1 strain may come out this time. Can we predict? Right? There are a lot of studies that are going on in this direction. How can bacteria acquire drug resistance? Where would you expect the change? These kind of things can be predicted. People are also using this to trace human evolution, migration, and genealogy. You must have heard of the IBM genographic project. And also things like 23andMe, you can actually send your sample and then get it sequenced and figure out which clade you belong to and so on and so forth. I think a service available for $100 or something like that, Okay, right. Okay, what do we get from all of that? You get a genome-wide annotation for each genome that is out there. This kind of an annotation is available. This slide is at least 10 years old, but it won't change too much except for this X marks. This is unknown, basically, that you see. Each square here represents a protein, and the color code tells you what sort of functional category it is. A broad functional category, this level, right? Now we have it for many genes, hundreds of genomes that is there, all through bioinformatics and all accessible. If you're working on some, let's say, Yersinia pestis or some such species, you will be able to just go and then pull out the information that is there and then That will be your starting point. Okay? You can also ask questions such as this. What makes Mycobacterium tuberculosis so different from Mycobacterium leprae? You know, both have the same starting name, so the same genus, different species. The diseases they cause are very different from each other, right? Now, this is a comparison plot, what we call as a dot plot. It's the same thing with different parameters. You can ignore that. So, on one axis, I put leprae, another axis, I put TB. Wherever there's a similarity, there's a dot. Wherever there's no similarity, it's a blank. Okay? That's interpreted for you in this kind of plot. This many genes of leprae are common between common with the TB. It's only this small portion out here that is different from that of TB. You can do the converse also. You can do, in fact, not just pairs, you can do across all of these kind of things. Okay, so you can do concordance, discordance type of analysis. Why is this useful? If you want to understand the differential pathophysiology, right, it may be this set of genes that will provide you a first shortlist. It's only a shortlist though. Okay, to say that because these are different in the two, maybe I should look into them and then see if they are in some way responsible for the differences in the diseases that they cause. Okay. So the next level of information, as I said, three-dimensional structures, typically determined by X-ray crystallography, nuclear magnetic resonance, NMR, and today also electron microscopy. So we have anything 100,000 structures available in a database called protein data bank, which gives you a structural or a high resolution view of biology. What do you do with structures? Like I said, same thing that you do with sequences, but much, much more detailed and much better. What you can do many things that you can't do with sequences also. Okay? For example, this is a structure of a protein that's important in DNA recombination and repair, RECA protein. Okay? I compare the E. coli and the Mycobacterium tuberculosis RECA proteins, and the 3D structure is the same at this level. This is a skeleton that I'm showing you, but there are differences in sequence, which we can map back onto this structure to see what is the implication of that, what's the consequence of that change. And one of the consequences of that change is a slight expansion in the functional region, something called a P loop, in the TB one as compared to the E. coli one. Okay? There's a slight, now think of it as a lock and a key. Now this keyhole is what we are looking at. Keyhole in TB is slightly larger than the keyhole in E. coli. What happens as a result? The fitness, you know, the tight fitness, the snug fitness that you see in terms of the ligand, ATP binding into that, is a bit more relaxed in TB, which makes it lower affinity, poorer binding, and more sluggish. That enzyme is much more sluggish. You can explain that. So it's just various ways. In fact, this is a representation of how this molecule folds over itself and forming filaments. Through this thing, the DNA binds, damaged DNA binds, and does its function. Okay, and this is E. coli, and this is E. coli, and this is TB. The color coding reflects the charge distribution on that. It's done through very sophisticated calculations involving finite element methods. Because we don't write the code for all of that, modules are available. You can do the calculation and see that the small differences here cascade into big differences when you look at the biological assembly of that. Okay, now details of the keyholes here and so on. We can also use it as an informatics approach and compare with all other skeletons of the similar kind that are available out there. See, what I show you is, we call it as a C-alpha trace, which is nothing but the alpha carbon atoms taken together. You superpose alpha carbon atoms of similar proteins across different organisms or whatever, and see how much of it is conserved, okay, which means what is the essential part of that. And now you can start predicting what is contributing to binding, what is contributing to stability, so on and so forth. Okay, and also what is the shape change of the ATP molecule, what are the conformational changes, and so on. Again, at the heart of it, we have physics models which understand bond length, the strain, the angles, and so on. I think I'm going a bit slow, so let me race a little bit. These are static models. You can also do molecular dynamics. In the sense, you can solve equations of motion to understand what is the best conformation of a particular thing, what's the most stable conformation, or does a protein change its shape through the course of its reaction, right? Those are the questions one can ask. Again, all computational methods, and get answers about what is the, unlike a rigid lock and a key, the keyhole changes, the key also changes slightly, sometimes reasonably highly. Okay, so you can understand, map all these contours, which becomes very useful for drug design. Now, instead of, this is some of our own work, instead of looking at one protein at a time, you can model protein structures of about 3,000 or 4,000 structures, proteins in Mycobacterium tuberculosis. You can do that for E. coli, you can do that for human. It's a tough exercise. I mean, it was one PhD thesis, but you can do this, right? It is possible to do all of that and get functional annotation at the level of structures. Again, you need to write a whole lot of new algorithms. Now, these are codes which will map three-dimensional objects, somewhat like face recognition, right? Or like contours, mapping contours of a geographic territory or something like that. It's similar principles, really. You are not only looking at geometry, but you also have to match chemistry. So you have to incorporate all of that into these kind of things. And once you understand that, so again, we are standing on the strength of physicochemical principles. You can see what sort of a change you must make to this substrate, which is ATP, to make this as an inhibitor. Now, I want to design a drug, right, for this particular protein. I know the contours of the keyhole now, which is our binding site. And if I start off by putting the natural substrate in and try and evolve it using algorithms which we call as genetic algorithms, I can end up with some molecule which looks like that but binds more tightly to this expanded hole but not to that other one. That's possible to do that. The challenges, but the many successful examples also. Okay. Again, most of these data that I said is available in databases which are cross-linked. You can understand, you can navigate across these databases and so on. Now, but is this sufficient, right? These are predictive models at individual levels. What more is necessary for us to answer bigger questions, basically? Again, hierarchical organization of living systems have talked about. So if you look at that, you need to be able to relate what happens in a molecule to an assembly, to an organelle, to a cell, and so on and so forth. So this requires the understanding of systems as a whole. Let me show you with completely another example which is completely different, right? So we understand to a major extent what are the changes that happen in diabetes, right? Diabetes mellitus, type 2 diabetes, for example. So what are the proteins that are involved are elucidated quite a lot. So we know the carbohydrate metabolism, various kinds of fatty acid degradation, a whole lot of other things, insulin signaling and downstream cascades, what are the various downstream effects of insulin and so on. Now you can ask a question, if you know all of this, these are just points put together, stitched together into your map. Can we ask a question, what is a good drug target for diabetes? No? Right? The answer is no. And have the drugs that are available, are the drugs that are available, utilizing the best possible strategy, right? And is the strategy the same in all patients of type 2 diabetes? So will a single formula, single drug work for all? These are the questions one can ask. In fact, it so turns out, we're quite fortunate, it so turns out that these 3 black boxes that you see are the drugs that are currently used clinically for treating diabetes. Okay, they happen to inhibit or mimic or whatever at one of these steps. They inhibit gluconeogenesis, one of them is a potassium channel blocker, and so on and so forth. This is a reverse pharmacology process. They were not designed with this in mind, they're designed differently, but they are useful. But there may be a whole lot of other things, some kinase inhibitors and so on and so forth, a large number of molecules. This again is a simplified map. To understand this, you need not just a static map like this, you need a mathematical model of the whole thing together to say, because it's at the end of the day, it's a game of numbers, right? You don't want to knock out one protein completely because that may be detrimental to the patient. You want to modulate it only to the level to correct it to some extent, the pathophysiology. So what sort of, which is the controlling point, for example, and how much should you modulate? These answers you will get only if you take the system as a whole and start modeling. So this is where systems biology actually comes into picture. Again, systems biology is not really a new discipline. Systems thinking has always been there. Okay, before we go into that, it simply means that the whole is greater than the sum of its parts. Now the genomics, the proteomics, sequences, structures, whatever we've seen are like giving you a very good definition of each part. So you can get a beautiful parts list like I showed you. But how to integrate them and how to make sense out of them. How to model the flow through this parts list. That's what we want to look at. So this is to understand how system properties emerge. That's the main goal of systems biology. Emergence is a key word out there. The pluralism of causes and effects in biological networks is better addressed by observing through quantitative measures multiple components simultaneously and by rigorous data integration with mathematical models. A pioneer in this field called Ove Sauer had said this basically. Okay, so again, to sum up what we are saying, a reductionist view, I think many of you would have heard of the terms reductionist approach and an integrated approach or a systems approach. Reductionist approach is taking it down into the most simplest bits and then saying that you can explain the whole thing by simply looking at the parts. Today, nobody really questions the fact that we can't have a reductionist view. But you can't start with the systems view and not have the detail. The detail comes from the reduction. So we need a merger of the two, really. So just to remind ourselves, actually, a reductionist view, this is an example of drug resistance, is to say that each one looks at an elephant. You've heard of the famous proverb, the blind man and the elephant. Each one looks at some particular thing and is very sure that is what it is, right? And to give you an analogy, we can have statistical significance of each one of these, say 10 to the power minus 23 or some such thing, so we're very sure what we are looking at. But when you put them in context, it may not be the same, right? So when you put it in context, so genomics, like I said, provides you this parts list. It's the systems biology which will give you this coherent framework to start putting things together, basically. Okay, in fact, it's predicted, I'm not sure of the actual years, but it is predicted that almost all biology in 2020 or 2030, doesn't really matter, will become systems because it's become more accessible today to start looking at whole systems. Okay, let me tell you what systems biology is not. Okay, now you're again familiar with the traditional experiments you would have heard or seen or done or something like that. You take a mouse, you inject something and record an output, right? You take a cell, you put something in and record an output. Is it using a systems concept? Most definitely yes. Which is why you don't rely on test tubes or in vitro models, but you want in vivo models. The reason why we need to have clinical trials before a drug is released is because of this, because there's so many factors that we haven't looked at. But quite often, in quotes again, this is a black box. We don't know why that output results. We only know what comes out. We don't know why and how. Those questions are not addressed. Okay? So systems biology is not that, it's different from that. Systems biology is also not this. This is on the other extreme. There's a joke among physicists about a spherical cow. In fact, a farmer goes the Midwest in the US, has huge losses and goes to the university thinking that's the cleverest place. Okay, the university summons the physicists thinking they're the cleverest. They go onto the field and record lots of observations, big notebooks, come back with an answer and call in the farmer to explain. The first line of the report reads, assume your cow to be a sphere in vacuum. The poor farmer is heartbroken and doesn't listen to the rest of it. In fact, it goes further to say you don't even need to assume it's a sphere, a point cow model works equally well. Right? This is making a joke about the extreme levels of abstraction that one needs to do in order to explain something. The generalizations at that large level, topical level. Not that they're not useful, they're extremely useful sometimes, but by systems biology is not that, especially molecular systems biology is built brick by brick from the bottom-up approach, from the genes onwards. No? So what you have here is you have a large amount of experimental data. Again, because you want the coverage to be as high as possible, it's not hypothesis driven here, it will generate hypothesis for you. So you need to look at everything, right? So omics data becomes important. And then you put them together into some sort of maps based on what is prior knowledge and so on, prior information. And you need to have it as a mathematical model. The challenge here is to convert a biological question into a mathematical abstraction of some kind. Right? You need to say what is important, what is not important, capture the essence basically. And then because it's so complex and you want to look at it, computational methods become very important. So the model has to be computationally tractable. You need to be able to simulate, get answers, verify. Verification or the validation is the first part of model building, after the model building is done. And only if it is able to rationalize what is already known, it becomes predictive. And then you can start asking a variety of questions which are useful for you can name it, it will be useful for that. Now, I won't spend too much time on that, but what sort of modeling you will use, again, not only depends on the hierarchy of the level of hierarchy, but also depends on what data you have, how much data you have, and what your question is. If you want answers, at what level do you want the answers, basically. The models, the taxonomy of models range from low granularity, means less detail, to high granularity. At this end, you have atomistic models at the atom level. But at this end, you have quantitative models, or machine learning comes somewhere in between, or actually spans across depending on what you have. So for machine learning, again, to understand what actually goes wrong in a disease, if you take the circuit of this kind, a network of protein-protein interactions maybe, something can go wrong in one protein or the connection between them, or it can go wrong in multiple proteins, right? We need to understand that. So let's see how we can. I think I'm running out of time. Is another 10 minutes okay? Is okay for all? Okay. So we want to model the phenotype from the genome. So that's a challenge. Take it through systems basically. So molecules to pathways to cells to tissues to humans. So what is the time scale we want to look at, and what is the spatial things? That's the kind of challenge. Not everything has been done. Also, although in the end I'll show you a physiome project where this is being addressed. Let's just start with machine learning approaches. Machine learning is now you don't really understand the basics, but you have a large amount of data. Say you can simply train the system and allow that to understand, right? Like how it's actually inspired by neural networks. Neural networks now today they have support vector machines. There are all sorts of things. This is an example of very complex data from cancer patients. You want to understand who is responding to treatment and who is not responding. Green is responding and red is not responding out here. So the data actually is transcriptome data. The gene expression profile of the same tumor type in multiple patients basically. You take all of that and simply try and classify in as many axes as possible, and you have a set. You already know a prior training set is available, who is responding, who is not responding. From that you learn which are the features which you may not be able to articulate by itself, but these are hidden features, and which are responsible for this classification. Once you get this classification access, you apply that on unknown data and see if it works equally well. And then you do not for 10, 100, you do for a large number, you get statistical significance. It becomes a more and more accurate model. So that's how machine learning methods work. So one is the support vector machine, but you also have various types of neural network and so on. Again, so this is to classify non-responders, responders, and so on. And you can profile patient data by this kind of a thing. So these work equally well for looking at sequences. Which are the sequences which are related to one particular function? Which are the structural skeletons which are related to your function? You can do it in different ways. The logic is the same. Okay. You can also do it for an area which is called chemoinformatics. A large number of drugs and their analogs are known. They're synthesized. You can look at small molecule features and say which portion of this small molecule is important for me to get a particular function. A function is inhibition. So it's not necessary to do it with this data. They are pretty agnostic to data. It doesn't really matter. So as long as you can convert the data into a form which can be queried, that will be sufficient. So like I said, most diseases are actually multigenic. You can have a perturbation here, a perturbation there, and so on. We can track in terms of networks, right? Again, an easy analogy to show you is the road networks, basically. There are roads and junctions, basically. We can model the protein interaction network. It may be much more complex, but we can still model it like a road network and now ask a variety of questions. Like to go back and say for Mycobacterium tuberculosis, I want to find out where to place a block such that I block the entire city's traffic. Which is the best, junction for me to place, junctions of proteins now, to put in a block. A block is an inhibitor, so that the entire metabolism or the bacteria just dies. This is the most important thing for that. That's a question that we can ask. Okay, in fact, we have answers for that. Something like, I won't go into the methods, but a whole lot of graph theory based methods with a solid mathematical foundation have been developed over the years that is being used. So in this jungle, you want to find just this path, basically, which is the most important for you. So some of these are from our own work, so these are from literature, basically. And you can use that to classify patients. In fact, this is an example from cancer. You can say, cancers arise due to many causes, right? Some due to mutation, some are silent mutations, some are expressing mutations, truncation, missense mutation, or copy number variation, amplification, all of these kind of things. It can come due to variety of things. Now, if you take all patients of a particular cancer and then start classifying based on this genomic data, they classify into some subtypes which may match with the known histopathology and other markers that are already there being used clinically, which will tell you what is the difference between one subtype and another. Okay, and then people, in fact, for breast cancer, it's pretty well known. People have found out what sort of a drug will work best for one particular cancer type, whether it's HER2 positive or not, and therefore give only that drug to that patient, right? And there are a lot of side effects which can be avoided. There are many advantages by knowing who will benefit from the drug and who will not. Okay, people have taken it further and say not only which drug, these are may not have entered the clinic as yet, but there's a lot of research on this, to say what is the order by which you should give and what is the best dosage of a combination of drugs that you can get out of that. Okay, and you can again go back to drug discovery and ask a variety of questions. Like I showed you this map before, we can capture this entire thing in one model, a file of 1 MB or 2 MB or something like that. You can capture this entire thing, which is what is an example from our lab about modeling mycobacterial metabolism, Mycobacterium tuberculosis. That entire thing I showed you has been put into something called a flux balance analysis model, and this is the result of the simulation of that. It becomes predictive now because you have modeled it and you have verified it. It's like playing a game, right? An in silico bacteria that you have, you can put in different types of food, carbon, glucose, glycerol, all of those things, and ask which is it happy to grow with, and is there a difference in the rate at which it grows? It becomes important, because mycobacterium is residing in the host and able to utilize fatty acids and other things as food. You can also change its eating rate, right? You can change the transporter rate or the flux of particular thing and then figure out how fast is it eating, and if you manipulate that, will you get a good result? And you can do systematic knockouts of each one of these and predict which is essential, right? All of these are totally essential because when I'm knocked out, I'm not getting any growth, no biomass in my theoretical model. Those kind of things you can do. You can also understand which pathway is linked to which other pathway and how much. This is a graph of pathway and so on. So this particular thing, membrane metabolism and fatty acid metabolism are heavily linked in Mycobacterium tuberculosis, but not in other organisms. So that's important for that. You can take it further and say process. Now carbohydrate metabolism as a process, if you put them together, you will see how beautifully it's orchestrated. Everybody is connected to everybody out here. It's a click, which means at the process level, everything is essential. At the pathway levels, you can have a rank list, and at the gene level, you can do away with some. They're not useful drug targets, but some are absolutely essential, which become drug targets. You can compare now TB with leprae and networks like this. You can do comparative network analysis as well. You can also put them together systematically, combine systems analysis, sequence, structures, everything, and at the end of it come out with high confident drug targets. Now you've generated a hypothesis which you can go back to the laboratory and test it and so on, right? Again, another example of how you can understand drug resistance. How is a bacteria acquiring drug resistance? Again, TB as an example. So use network data, transcriptome data, everything, and find out what are the major variations in this road network in drug resistant versus drug sensitive conditions. Understand these things and pick them, and then you will get, in fact, a combination of drugs which can reverse resistance is what we've identified. And there are many examples of this kind, right? Okay, so let me skip this in the interest of time. So it's also useful for biomarker discovery. See, if you can find out drug targets, you can also find which are the good telltale characteristics of this disease that you can find. You can do that in the host. You can also do that in the pathogen, basically. Again, let me skip this. The last example that I wanted to tell you is you can also understand who will mount a good immune response against, let's say, a viral disease such as influenza virus, right? By understanding the immune components and then sequencing and their structures and taking it forward like that, this is a whole number of people here, models, all models here, are grouped into only a few response types. Okay, so each set of people, it's about 1 million people basically, and here this set of people based on their HLA makeup, the HLA genotype, human leukocyte antigen genotype that an individual has, can be grouped into only a few types. So each type here will see a pathogen in the same way. That's the prediction. Okay. So and then if somebody has that particular HLA genotype and a particular strain of virus is going around, you can predict what sort of a response they will have and whether they are susceptible to disease or not. That's the kind of work one can actually do. So here these are all H1N1 influenza strains, and these are all various ethnic groups that are modeled. And the height here shows you how much T cell, CTL, cytotoxic lymphocyte response you can have, CD8 response, how much you will not have. The more the response, less the susceptibility and so on. So you can do this kind of analysis, it becomes predictive. So it takes it all the way from metabolic networks, disease networks to social networks, and you can also predict disease spread by different types of models. Now combine epidemiology with this and find out how fast an infection can spread, which will give you some idea for vaccination strategies, who should be vaccinated and who need not be vaccinated. So where are we heading? We're heading towards virtual physiology, basically, understanding. Again, I think we'll run out of time, so let me just skip the example of the Physiome project that I wanted to talk about. So ultimately, for any model to become useful, we need to see coherence. It needs to be a coherent model, which means it needs to be able to explain various disparate experimental results that are out there, stitch them together and make sense. That is what it is. And the beauty of systems comes from emergence. In fact, it is said the simplest living atom is actually a cell and not an atom, right? Way back in 1860, somebody said that. And Monod has said that living structures, there's no living matter, it's only living systems. So systems become very important to see. I think I'll leave you with this quote, which says ante, not anti, ante-disciplinary science, which means that before discipline. So you don't really need disciplines. It's actually why computation and why next generation biologists must straddle both computation and biology, basically mathematics, everything. So you need, just because most of you are from biology, you should not think that it is not important for you to understand other areas. You need to have an integration of various things today, and it's becoming quite important to explain biological processes at the level of various things, right? And yeah, thank you for your attention.